Cystic fibrosis is a genetic disease that predominantly affects the lungs and the pancreas. But what exactly is it and what is the cause? Cystic fibrosis is autosomal recessive. This means in order to inherit the disease, the mutation has to come from both the mother and the father, so both of the genes in the child are the mutation. There are recessive alleles and dominant alleles. A dominant allele covers up the effects of the recessive allele, so unlike recessive alleles, you only need one dominant allele to show the dominant trait. In this Punnett square, both the mother and the father are carriers for the CF gene. In the example, there is a 25% chance that a child will not have the CF mutation. There's a 50% chance that the child will be a carrier, and a 25% chance that the child will have cystic fibrosis. Cystic fibrosis is caused by a mutation of the CTFR gene, which is found on the long arm of chromosome 7. This gene encodes the CTFR protein. It is a protein that works as a chloride ion channel for ion transportation in and out of the cell. This ion channel controls the concentrations of salt and water inside the body. But how does this mutation create cystic fibrosis? Inside and outside of the cell, there's a bunch of water and salt. The salt is formed from the chloride ions when they combine with sodium. When there's a functional CFTR protein, the chlorine ions will enter and exit the cell and then merge with sodium ions where they will form salt. Through osmosis, water will follow where the salt concentration goes. In summary, where the chloride ions go, so does the water. In cystic fibrosis, where the CFTR protein is deformed or not even there, then the chloride ions will get trapped inside the cell, making salt concentration inside the cell high, preventing the water from osmosing out of the cell. Cystic fibrosis predominantly affects the lungs and the pancreas. In a lung with a functional CFTR protein, water outside the cell thins out mucus. This makes it easier for cilia inside the lung to push mucus through the airways. In a lung with cystic fibrosis, there's not enough water outside the cell to thin out the mucus. The cilia inside the lungs then have a hard time moving the thick, sticky mucus throughout the airways. When a healthy lung breathes in a pathogen, the pathogen gets stuck on the mucus. The cilia then move the mucus and the pathogen out of the lungs. In a lung with cystic fibrosis, the pathogen sticks to the mucus. But since the cilia cannot push the mucus and the pathogen out of the airways, the pathogen ends up infecting the lungs. There can be many pathogens stuck in the airways, meaning there could be many lung infections. Other CF symptoms inside the lungs include trouble breathing and persistent coughing. Most CF patients die from these lung complications. As patients grow older, the complications get worse. The average lifespan now is about 44 years. Back in 2000s, it was about 20 years old. In a pancreas with a functional CFTR protein, digestion enzymes made in pancreas cells leave the cell and go through the pancreas passageways to the stomach to help with digestion. In a pancreas with CF, the passageways in the pancreas get blocked with thick, heavy mucus. The pancreas enzymes build up in the pancreas cells and don't reach the digestive system. This results in difficulty digesting, greasy, bulky stool, frequent difficult bowel movements, and an inflamed pancreas. There are 6 classes of CFTR mutation for cystic fibrosis. In class 1, there is no protein synthesis inside the nuclease, meaning there is no CFTR protein. 22% of cystic fibrosis patients have this mutation. In class 2, there is a block in processing in the Golgi body. An abnormal CFTR protein is made. 88% of cystic fibrosis patients have this mutation. In class 3, there is a block in regulation. 6% of cystic fibrosis patients have this mutation. In class 4, there is reduced conductance with the CFTR protein. This means the channel works a little bit, but not as much as it should. 6% of cystic fibrosis patients have this protein. In class 5, there is reduced synthesis, which means they're all functional CFTR proteins, just less of them. 5% of cystic fibrosis patients have this mutation. In class 6, there is a functional CFTR protein, but it has a reduced half-life, meaning the protein doesn't last as long as it should. 5% of patients have this mutation. Many cystic fibrosis patients have more than one of these mutations. A treatment for cystic fibrosis includes CRISPR-Cas9, which is a gene editing tool. CRISPR-Cas9 is naturally found in prokaryotes like bacteria. CRISPR stands for clusters of regularly interspaced short palindromic repeats. They are specialized regions of DNA with nucleotide repeats and spacers. Spacers are viral genomes from past virus infections. After a virus attack in bacteria, viral DNA is incorporated into the bacterial genome to protect against future attacks. Cas9 is a CRISPR-associated enzyme. It is a nuclease. They are molecular scissors that cut DNA at a specified location. To add the viral genome to the bacterial genome, Cas9 is what cuts the bacterial chromosome to include the viral genome. Guide RNA is used in the gene editing process. It binds to Cas9 and tells it where to cut the DNA. The repair template is then the correct genetic sequence for the mutated gene. It replaces the incorrect sequence, the mutated sequence. Now let's go over the gene editing process step by step. DNA is stored in the nucleus. The red section is the mutated CFTR gene. The guide RNA binds and activates the Cas9. Cas9 then searches for DNA that matches the guide RNA. Not shown here is Cas9 recruiting helicase to unwind the DNA so Cas9 can cut the strands of DNA individually. Once the mutation is cut out, the Cas9 is inactivated, and the template strand is introduced. DNA polymerase then comes in and uses the template strand to make a complementary strand on the DNA. Then the DNA polymerase makes a complementary strand to the newly made strand. The DNA rewinds, and that cell now has a functioning CFTR protein. There are limitations to this gene therapy. There are over 1,700 CF mutations, which means in order to use CRISPR-Cas9, you have to pinpoint which mutation there is and make a guide RNA identical to that mutation. Also, Cas9 can cut in the wrong place, resulting in more mutations, even cancer. CRISPR only works during DNA synthesis, which means lung cells that do not divide cannot be fixed. Also, it can be difficult to get gene editing tools inside of critical, hard-to-reach organs. There are a few ways, though, to deliver those gene editing tools inside the patient body. Gene editing tools can go in liposomes, which CF patients then breathe in via a nebulizer. Gene editing tools can be made as a virus's genome. That means the Cas9 and CRISPR and all of that is the virus's genome. The cystic fibrosis patient is then infected with the virus, and the virus then inserts the DNA into the cells, where gene editing then takes place. The downside to viral gene delivery is that few viruses deliver DNA to the lungs. So, where cystic fibrosis is a big problem, no gene editing will be happening. Though there is no definite cure today, hopefully tomorrow there will be one. Till then, this treatment helps cystic fibrosis patients live longer and easier. Thank you.